today's Chardan leadership call featuring Cabaletta Bio, a company focused on developing cell-based therapies for B-cell mediated autoimmune diseases. A special thank you to our guests, Dr. Steven Nichtberger, co-founder, CEO, and chairman, as well as Dr. David Chang, Chief Medical Officer, and Anut Marta, Chief Financial Officer of Cabaletta Bio. I'm Geulah Livshits, a biotech analyst at Chardan, and as call participants are aware, our focus at Chardan is on identifying companies that can generate exceptionally high long-term investment returns by creating and sharing in value for society. During this call, we will not be discussing Chardan research. Any discussions about Chardan research should be coordinated between participants and their respective salesperson at Chardan, and a recording of this call will be made available to clients afterwards. Our compliance team has asked me to read the statement for the call before we get started. So by participating, our speakers attest that they have made Chardan aware of any potential conflicts, and they will not discuss any material, non-public, or confidential information that they're aware of that may breach their legal, regulatory, or fiduciary responsibility to any parties. In terms of format, this is a 45-minute to 1 hour long webinar, and participants can ask a question. To do so, enter it into the Q&A box in the Zoom window. So thank you all for joining us today. And to start off, Steven, would you give us a high-level overview of Cabaletta as a company and its history for those who might be new to the story?
Absolutely. And thanks for the opportunity, Geulah, to share our story with some of the investors. And thanks for investing the time with us today. So the company was formed in mid-2018 in order to develop and launch the first targeted curative cellular therapies for patients with autoimmune diseases. And our target continues to be deep, durable responses that are perhaps curative, but we don't necessarily need to achieve cure in order to achieve a great outcome for patients and for our investors. So our eye is on the ball. Our eye continues to be on curing these patients, providing at least a deep and durable response. The technology that we're using is a proven technology platform. It's the CAAR-T product design that was used to develop Kymriah, and the signaling chain that was used, the manufacturing process that was developed at the University of Pennsylvania, and the standard operating procedures associated with that are all part and parcel of our more targeted product, whose only difference from the Kymriah effort at Penn is that our targeting domain is not an antibody fragment to the CD19 antigen protein on a B cell or on a cancerous B cell as well. Our targeting domain is the antigen that's being attacked incorrectly in an autoimmune disease. And in the case of our lead program, pemphigus vulgaris, those patients have antibodies, autoantibodies that are pathogenic, and they are attacking the DSG3 antigen, which is essentially skin glue, desmoglein. That skin glue is being attacked by the antibodies in the patient. That leads to the skin having blisters, which prior to the use of high-dose steroids and other broad immunosuppressants would have led to death in the patient. And now it leads to a revolving door of treatments and relapses over the course of a lifetime in the vast majority of patients who have the disease. We chose this as our first target because it is, like all of the targets we have spoken publicly about in our portfolio, it is the prototypical example of an autoimmune disease where the B cell is at the center, where the B cell and the autoantibodies are necessary and sufficient to cause the disease. If they are necessary and sufficient to cause the disease, their elimination should lead to a deep and durable, perhaps curative response. And it's on that basis that we formed the company in order to prosecute this first-in-man study. But more importantly, that first-in-man study, we believe, has very clear and direct read-through to a very deep and only partly announced portfolio. So we're very excited that our initial clinical experiences from our clinical trial at the very low doses that we started, the 20 million cell dose and the 100 million cell dose cohort, very excited that those seem to be extremely safe. No dose-limiting toxicities in the 28 days after infusion. More importantly, no clinically meaningful adverse events of any sort in any patient at any time until last we reported publicly, August 17th of this year. We are, as we previously have said, moving ahead in the dose escalation. And as of August 17th, we said we had started dosing in that cohort. And as a reminder, Geulah, just to close out this introduction, we are announcing clinical data on a cohort by cohort basis, with the key timelines being 1 month after the third patient in a cohort, or the final patient in a cohort, is dosed. We will talk about, or we will have in our hands, I should say, and then subsequently announce the 28-day acute safety data. And then between 3 and 6 months after that last patient is dosed in a given cohort, we'll have the full complement of data on early signs of target engagement or clinical efficacy, and we'll be in a position to talk about those. We expect later this year the third cohort, which is a 500 million cell dose cohort without lymphodepletion. A 500 million cell dose cohort will be able to report the acute safety by the end of the year, and the target engagement or early signs of clinical efficacy, that data will be reported on the first 2 low-dose cohorts by the end of the year.
Great, thanks for that great overview. And more broadly, are there any other catalysts that we should be aware of that are anticipated over the next 12 to 18 months before we dive into the programs in a bit more detail?
Yeah, 12 to 18 months, my brain is on fire because there's so much going on. So let's just stick with the next couple of weeks. So, and then I'll take you beyond that. So the MuSK IND, our second program into the clinic, is very much on track for filing, as we've said, this year. We are adjusting the timing of that IND filing to accommodate and to incorporate all of the learnings from the DesCAARTes trial in our pemphigus program. And that's really important because the first to the starting line is not necessarily the first to the finish line. And our real objective is to be across the finish line first. And in that regard, whether or not we use a 20 million cell dose cohort in the MuSK program will partly rely on seeing the target engagement or the efficacy data in that cohort. So waiting to see that data might be the wise thing to do when it comes to the MuSK IND. Similarly, whether we use 4 fractions, which was initially designed into our trial, our dose escalation portion of our trial, for different fractions, starting at 1%, going up to 69%, so that the total 100% of doses administered within a week. That approach was because we first to do no harm. We now have, increasing numbers of patients, increasing dose levels with, extremely strong safety profile. And if that's the case, do we request of FDA that we move ahead and, with fewer dose fractions, for example? So there's a lot going on the MuSK program, but the IND will be filed this year. And that's for MuSK myasthenia gravis, where even Soliris is not effective. So the program after that is membranous nephropathy directed, PLA2R membranous oriented membranous nephropathy, another disease where the B cell is at the center, where kidney failure can be expected in a substantial portion of the rituximab failures who show up every year in the United States. It's about 1,000 patients who don't respond adequately to rituximab, and a very substantial portion of those patients will go on to renal failure, needing dialysis or transplantation within a couple of years. If we can potentially eliminate those antibodies in a selective and specific way, allowing those patients to avoid the need for dialysis, avoid renal failure in a very enriched way, not just a subtle population-based effect, but in a very enriched way, we see that as incredibly high value for patients and payers, and realizing ultimately the value proposition of the business.
Great. That makes a lot of sense. And so in terms of the platform itself, you gave a great overview of how it works and similarities with the CAR-19 program. Can you elaborate a little bit more on the biology of the CAR-T platform? And also, for example, there have been some recent publications and reports of B-cell directed CAR-Ts showing some efficacy signals in other autoimmune diseases, not in a targeted way, but in a broadly CD19 or BCMA specific way. Maybe you can comment on the read across that might be seen from those type of studies to your program, and also highlight any differences that we should be aware of.
Yeah. Well, the differences between us and everybody that we know of start with the fact that we are, biologically highly specific. It's the best description I've heard over the years of our platform is that it's elegant biologically. The B-cell receptor that is required to be present on a B-cell in order that it knows where it is. It knows when it bumps into the antigen that it seeks to attack. That antibody, which is the B-cell receptor on each type of B-cell, that antibody on the surface is the target for our T cell, and that antibody only exists on the 1% of your B cells or fewer that we seek to destroy. That sort of elegant biology allows us to believe that, at first to design, and then second to believe based on our data, that we should be able to eliminate only the 1% or fewer of your B cells that are causing your disease, leaving you with a normal immune system. In a post-COVID world, that's a really big deal. It was always a big deal. But now when you start thinking about the risks of COVID in an immune compromised patient, it's a whole different story. And so the Pemphigus Patient Foundation has a very active group of folks. And at a recent conference, the work that we presented and our Chief Medical Officer, David Chang, presented our clinical trial design and the early safety findings, which have previously been disclosed. And that oral presentation was voted as the top oral presentation of that entire scientific conference. So what we're doing is simple, it's biologically elegant, it's potentially clinically curative, and it's got profound value for the healthcare system, far beyond even just the patient benefit, because the alternative for these patients is years, if not decades, of increasingly expensive and only modestly effective therapy that comes with a boatload of side effects. The first of which is, in almost all cases, steroid boluses are used and chronic steroids are required. So people talk about rituximab being an important therapy in patients with pemphigus. If you read the label and you look at the papers, what you realize is the average patient is getting 3,500 milligrams of steroids in order to achieve, on an annual basis, in order to achieve a potential, a 40% chance of getting a 4-month hiatus from their disease off of all medicines. The vast majority of those patients, we know from published and yet to be published data, are going to have a recurrence. And so cure in a highly targeted manner is what we're shooting for. And we think that distinguishes us from every other therapy that we know of, either on the market or being developed. The fact that cell therapy can work in autoimmune diseases is profoundly important, right? So the fact that other forms of cell therapy are starting to see some level of treatment effect, perhaps, is important. It's important information. But at the end of the day, we don't buy into the belief that it's acceptable to leave a patient with B-cell aplasia, nor is it acceptable to expect that you're going to re-administer therapy on an annual basis or even more often in order to keep the disease at bay, right? If you're going to go for the jugular and you're really going to try to help these patients in a serious way, you want to get rid of those B cells once and for all and do it without side effects. I can't tell you that we have efficacy yet, but I can tell you that at the doses we've studied and reported, we have extremely strong safety. Great,
that makes sense. And so I think you covered the benefits and the differentiation there as well. So maybe we can move on to the DSG3 CAR-T program and talking about that in a little bit more detail. So you gave us a bit of an overview on the mucosal PV as an indication, but can you perhaps elaborate for us regarding the unmet need, in particular in terms of the kind of efficacy signals that clinicians and patients are looking for to move the needle versus the available care?
Yeah, let me ask David to step in here to respond. And we've publicly talked about 4 parameters that we're looking for as the early signs of efficacy signals. But let him walk you through in more detail as he's much closer to it and much more knowledgeable.
Yeah, and there's the one set, which is we're looking at the early signs of biological activity that we would be able to report on. So those would include probably earlier versus later, something similar to what we've already reported. Persistence of the DSG3 CAR-T cells, that would be the early biological indicator. Maybe something that would follow a little bit later, but probably just as important is looking for the drop in the DSG3 autoantibody levels, which would be an indication that the B cells have been ablated. And then subsequently, probably more important for patients is actually, did I get better? Is there evidence of clinical efficacy? Did the lesions go away, and did I not get any more further lesions? And also important for patients is, do I need to take any additional therapy? Do I need some rescue medication that needs to control this disease, such as steroids, or do I need to take additional immunosuppressive agents? So those are 4 parameters that we're focused on in terms of biological activity indicators. But when you think about what do patients look for, as I mentioned, they want to feel that they have gotten a complete resolution of their symptoms, and they don't need to take another pill or shot again. So durability is quite important for these patients, that they do not want to be on chronic therapy, especially therapies that may lead to immunosuppression and putting patients at risk for getting an infection or being prone to not getting a response to a COVID vaccine, for example. And the third thing patients really want is, related to that one, is they don't like steroids. Steroids is a double-edged sword. On the one hand, it helps them, but none of them want to be on steroids forever, right? For that matter, even for a short period of time, just because the multiple side effects associated with steroids for patients. So those would be things that we're looking for, and those are things that patients are looking for.
Great. And in terms of the trial design for the ongoing study, can you walk us through the different components there with respect to the enrollment criteria and the structure of the dose escalation that Steven alluded to earlier, and the consolidation and dose fractionation and those elements?
Sure, yeah. What we've designed is actually an ability to involve a broad patient population. So we're not taking a very, very narrow scope of homogeneous patients with only very specific type of disease. We think that the mechanism of action of our therapy is amenable for all types of patients. So this includes mild, moderate, severe, from refractory to rituximab, not rituximab naive, et cetera. So what we've designed is a study inclusion-exclusion criteria that only requires the evidence of active lesions. So doesn't have to be super severe, but that these patients have some evidence of clinical disease activity. And secondly, we're looking for patients who have at least tried one prior therapy and had either a relapse or intolerance or inadequate response. So we leave the door open for a very broad patient population to enroll in this study, which ultimately may translate to a label that could be just as broad once we get to that stage. So that's the inclusion-exclusion criteria. But the other thing I do want to mention with regards to exclusion is that we don't want patients to have recently taken rituximab and continuing to get better, because rituximab recent use could confound the results. And also rituximab use, and the patients have very little or no B cells or lower B cell count, we would lose our ability to be able to actually see whether our drug has a response to patients who have pemphigus or not. So recent rituximab use in which patients are actually getting better or have gotten better would not qualify for the study. And we don't want patients on really high doses of steroids, and not necessarily because we don't think they would respond, but because if they're on a very high dose of steroids, those steroids could potentially suppress the activity of our CAR-T cells. So we do ask for a ceiling on what amount of prednisone they should be on or lower, and that ceiling is approximately about 0.25 milligrams kilogram per day. So for your typical adult humans, that would be 12, 15 milligrams per day. And the study design, the way we have it set up is with 3 different parts. There's a part A, part B, and part C. With part A, that's the dose escalation portion. So we started off with a very low dose, a dose that probably, which Stephen calls the mouse dose, and it probably is, at 20 million cells as the low dose. Because remember, this is primarily a safety study, and we don't want to run into a major issue with the first cohort of patients. So we designed this in a way to be very cautious, start off with a very low dose, and then escalate. And the escalation, you've seen that in our press release, that we start off at 20, go to 100 million in the second cohort, followed by 500 million in the third cohort, and 2.5 billion in the fourth cohort. And if you've done the math, that's a 5-fold increase from A1 first cohort to the fourth cohort, A4. But it does span a very broad range from a very low mouse dose to dose that's been probably much higher or moderately higher than what's been used in oncology, with oncology doses probably being a range of 200 to 500 million cells for most of the oncology studies. So we're spanning that range within the oncology one and going lower and going higher as well. The other point I would make regarding the dosing regimen in part A is that we're using a fractionated dosing scheme. So instead of giving one large infusion, 20 million, 100 million all at once, we split it into 4 separate infusions. And again, this is primarily to mitigate safety issues. And instead of actually giving 4 equal doses, what we've done is to use a graduated escalation within the patient. So we used 1% of the total dose first, followed by 5%, 25%, and 69% or rest of the infusion on the 4th infusion. And this is generally given sequentially over 4 days. And if there's a weekend, then we would have to bridge over the weekend if they're not in infusion then. But again, this is really for safety. So start low, 20 million, and fractionate it with 1%. So the first infusion of the first patient got 20 million cells, but 1% of that. So it's a very, very low dose was given for that particular patient. And that was done, same thing for the first 3 patients in that cohort. So as Stephen has mentioned, we've already gone through and dosed the first 2 cohorts and have followed them out at least for 29 days. And noting that there's no evidence of DLT, no clinically relevant adverse event reported on any of those 6 patients for that 28 days following the infusion. And we've also reported that we have started looking into the 3rd cohort. So that's the 500 million cell dose range. So that's where we are today. The second part is part B, where we try to consolidate the dose. So instead of 4 separate infusions, we try to bring it down eventually to 1. So in a graduated step, so there's a B1 cohort and a B2 cohort. And if things go the way we think it will, we will have a consolidated single infusion, 100% of the dose by the time we get to by the time we finish the B2 cohort. And cohort C is just a expansion, 12 additional patients at the dose and the fractionation that is found to be tolerated and acceptable. So that's basically the design in terms of inclusion, exclusion, as well as the dose escalation in the 3 parts.
Mm-hmm. Got it, great. And so the current cohort and set of patients doesn't use conditioning. Can you explain why this is important and how that might be a lever that could be adjusted later on if needed?
Yeah, I'll start off, and then Stephen, if you want to add anything. So preconditioning, we said, well, what is the rationale that there's preconditioning or lymphodepletion that's used in oncology? And when we looked at all the rationale, it's to create space, it's to remove the cytokine sink, It's to remove the suppressor cells or regulatory T cells. When you look at autoimmune disease, it's actually quite different in the sense that we don't deal with that same issue. We don't have to debulk anything. We don't have to create space, because otherwise these patients have a normal cellular environment. They just have some abnormal B cells that are not behaving. So there's no debulking required. There aren't a whole lot of regulatory or suppressor cells that we have to worry about. And there's not these billions of other cancer cells, sucking up the good cytokines that would maybe needed for CAR-T cell expansion. So when we thought about that, we said, the first approach should be to follow the science. And we said, there's no rationale to actually give chemotherapy preconditioning prior to giving our CAR-T cell therapy. So that was the main rationale. I mean, there's other reasons we could add to that, but that's really the science answer. And really for the patients would say, you're going to give chemotherapy to me so I can receive this therapy. For oncology patients, that may be acceptable. For dermatology patients, our first approach would be not to do that. If we do need to do that and we can provide a cure, we'll explore that if that's something we need to do. But for the first approach, we would like to, our approach has been, let's follow the science and do what's most appropriate for patients. And we thought that this is the approach that we would be taking. And so far, so good. And we'll find out as the study progresses.
Yeah, I think there's a additional point that David has made before, and I'll offer to make it now, which is the soluble circulating antibodies are absent in cancer. You don't have that opportunity, but in the patients who we treat, there are soluble circulating antibodies. Our preclinical studies, which dove very deeply into the impact of the soluble circulating antibodies, revealed that patients with soluble circulating antibodies, their serum, when exposed to our product, causes our product to proliferate. Well, wait a minute, that's exactly why you give preconditioning. So it's very possible, if our preclinical data bear out in the clinic, that when we get to adequate doses, whatever that number would be, we're going to see product expansion by virtue of the patient's own milieu, the patient's own soluble circulating antibodies. So that opportunity is one that we're taking advantage of by not giving the preconditioning chemotherapeutic regimens and instead relying on biology. And again, as David said, just following the science. So look, we'll see what things, what the data say about the biologic activity, the target engagement, and so forth. We can, if we need to, add a touch of cytokine enhancement or chemotherapeutic preconditioning, but it's not without. We wouldn't go there without first exploring the more logical and what we believe likely to work scenario, which is what we're doing now.
Got it. Great. And so Steven talked a little bit earlier about the endpoints in the study. I don't know, David, maybe you could elaborate a bit about the safety and activity endpoints, and in particular the rationale for the time points that are used to assess the safety as well as the biologic activity.
Yeah, the safety endpoint, looking at the dose-limiting toxicity and to determine the maximum tolerated dose, that's based on, we went basically the way that oncology studies have designed it, which is a 28-day period post-infusion. And so that's a very conservative approach, because we know that most cytokine release syndrome, as well as neurotoxicity, probably occurs within the first few days of infusion. But just to be conservative, we did follow the same timing frame that oncology studies have done, which is 28 days for the dose-limiting toxicity safety endpoint. With regards to the efficacy endpoints, this is not an efficacy study, but we are looking at a variety of endpoints throughout the study, whether it's looking at the drop in autoantibody titers, but also looking at clinical scores, use of concomitant medications, including steroids, et cetera. So that's just being collected throughout the study. But I think maybe I'll add on to that, the time that you saw regarding 3 months or 6 months, we did talk about 3 to 6 months as being the time when we might start to see some evidence of biological activity as regards to autoantibody titers. And the rationale for that is that even if we eliminate all the autoreactive pathogenic B cells with our therapy, there's still the circulating autoantibodies that need to be metabolized out. And with the half-life of those autoantibodies being 3 to 4 weeks, you need to have a certain amount of time. And we thought 3 months, just by calculation, seems to be a reasonable time to start seeing a drop or evidence of biological activity. 3 months being the earliest, but ranging between 3 to 6 months.
Got it. And I know you talked a little bit about the safety profile that is disclosed on the program thus far. In terms of risk, from your perspective, were the biggest sources of risk the typical ones that you think of in relating to CAR-T therapies, or was there anything else that you were initially concerned about that might be specific to this platform or program?
Yeah, I mean, I think number 1 and 2, Steven, I'm taking your words.
Yeah, no.
Number
So
1 is
look,
CRS, right?
when you think of CAR-19 therapy, and you talk, as we do, to literally hundreds of investors and physicians, and you ask, what are the side effects of CAR-19 therapy? The answer, of course, rolls off everybody's tongue: CRS, neurotoxicity. And then you say, okay, what's the third thing you're worried about? No two answers match on the third one, because there is nothing. There's nothing that is a prominent side effect that's clinically meaningful that you can expect to see in some portion of patients who get CAR-19 therapy. And so it's our belief that those are the two major side effects to navigate across. And it's our belief that the number of B cells you kill is directly related to the risk of CRS and the severity of the CRS that you'll see. So biologically, we never believed we would run into any CRS or neurotoxicity. But when you're, being a physician myself, and when you're trying to do no harm, which is, of course, David's, number 1 priority as the leader of our medical efforts, you assume that you don't know what you don't know. And so you start with a dose fractionation regimen using what I call a mouse dose, 20 million cells. And you go through that process and you look for biologic markers that maybe there is no clinically evident CRS, but maybe biologically there's something brewing, or who knows what. You do everything you can to find that side effect. And, here we are many, many months after the first patient was dosed at the low dose. And there's not a single iota of any side effect that is clinically meaningful in any of the 6 patients we've spoken about publicly. To repeat, we've now, as of August 17th, started dosing at 500 million cells. And some people say, oh, that's cancer dosing. Well, not quite. We're not using lymphodepletion. 500 million cells in cancer doesn't work unless you lymphodeplete. Our belief, based on the BCMA data that we've reviewed, Geulah, with you and others previously, is that when you get up to substantially higher doses without lymphodepletion, there is evidence in BCMA-directed CARs that you can achieve similar clinical outcomes without lymphodepletion as you achieve with normal dose CAR-T with lymphodepletion. Well, the problem is you can't go to very high dose without lymphodepletion in cancer because it's too toxic, CRS and neurotox. But if you have a therapy that doesn't kill a lot of B cells, and arguably we kill between 3 logs and 7 logs fewer B cells than cancer CAR-T. When you talk about that kind of dose separation, that kind of headroom to a side effect, you can go to much higher doses than are commonly used in cancer. And we expect that as we do that, we're going to get deeper and deeper responses that are more and more durable, and we're going to realize the promise of the company and of our platform. So, navigating across the first 2 dosing cohorts, admittedly low dose, and having this safe profile is not only encouraging, but exciting for us. Because, at our IPO, we talked about, and those who were there and who received a presentation from us in that period, you'll know that there was a slide that any recorded presentation of me is going to have me saying, if you don't pay attention to this slide, which lists all the things we're concerned about, all the safety risks, then don't invest, because we really don't want to hear that you didn't know. And at the end of the day, maybe a lot of people listen to us, and that's fine. We're doing the same thing now on the efficacy side. Before we saw any efficacy data, we started thinking about, if we don't see efficacy with a sufficient number of cells, what are the 6 or 7 different opportunities we have to get to cure? If we don't see an adequate response with just these 4 arms of this study, what are the options we have to migrate into more effective therapeutic doses or applications? And there are many different opportunities. We haven't spoken publicly about any of them yet. Just like the safety risks that we mitigated, and to this point, we've never had to pull those tools out of our toolbox. On the efficacy side, nobody should read anything into the fact that we are planning for the challenges that we don't even know we have yet. We wouldn't be doing our job if we didn't plan for it to be hard. And so in that context, we've got a large number of opportunities, and to throw out with no particular preference or implication that we would go here first, second, or never, a low dose of a cytokine enhancing regimen, either in the manufacturing process to make the cells more potent, or in the patient, a low dose of a chemotherapeutic agent, one of the preconditioning agents that are used. There are just so many different opportunities that we have to get to efficacy, enabled by, permitted by the fact that the therapy itself seems to be as safe as the biology would have suggested it should be.
One thing I could add to that is that one thing we do know, and we've put this out in our corporate slide deck, is that with the first patient, we were actually able to take that person's manufactured product and do an in vitro assay and say, does it do what it's supposed to do? And we actually combine those T cells, the CAR T cells, with target B cells that are expressing the B cell receptor, the anti-DSG3 autoantibodies, and the cytotoxicity assay worked. And there's a dose response. The more cells you give, the increase the target effector ratio, the more killing there was. So we know that manufactured product will do what it's supposed to do in the test tube. And what we're at now is, let's see how it works in the human being, because there's more variables. But that's why we have these other efficacy parameters, opportunities that we're looking at. But I think we felt very comforted by the fact that we know that these manufactured products do what they're supposed to do in the test tube.
Yeah, Geulah, you asked early on, what's coming in the next 12 to 18 months. I think our firm belief is the sort of deep, durable response, the sort of efficacy that we expect to deliver from the platform is what we hope is coming. And the way we get there is complete dose cohort number 3, complete dose cohort number 4, review our safety data in both cases, look at the target engagement, the biologic activity in the 3 to 6 month window after each, and well before we get to that point, reveal our plans for any subsequent dosing regimens or approaches, because we do intend to continue to raise the dose until we see toxicity. And the reason for that is very simple. It comes back to the very first thing we said on this call. We're shooting for cure. That means deep and durable complete response. And the only way you get there, in our view, is maximize the opportunity for this platform to eliminate 100% of the B cells and to do so permanently. And it would be short-sighted of us to declare a victory until we have given the maximum regimen that we can give to patients in order to believe that we have the maximum durability, which you can't know your durability until years from now, right? So it'll be very thrilling to have the early efficacy data, right? Which will be profoundly different from anything else out there in our view. But the durability is, at the end of the day, where, for the patient, the real value proposition comes getting to a maximum effective dose, which is the purpose of our study, is what we're trying to shoot for. So the first 4 dose cohorts are out there in the public domain. It's part of the announced protocol. There's much more after that. And again, I'll reiterate, nobody should read anything into what we might see in the 100 million cell dose cohort, the second of the 4, because we don't have that data yet. And we're not planning for failure. We're just planning because that's what you should do when you're running a study like this.
Great. I think those are really helpful insights. And on the topic of durability, this might be a more philosophical question at this point, but in terms of potential durability, do you think that you need to have persistence of the CAR-Ts to have durability in this type of indication, or is there reason to think that even a potentially transient or for some number of days or months peak of the cells might be sufficient to induce a durable remission?
So the answer is, in humans, we don't know. That's why we're doing the study. Is it comforting to have seen persistence within the first 28 days, during those first 28 days in all 6 patients, despite an extremely low dose? Absolutely. If you look at cancer and you ask, is persistence required? Among complete responders reported out of the University of Pennsylvania in a letter to the editor in New England Journal was this year or last year? With COVID, every day is the same. So within the last 6 to 8 months, I believe, they reported that among all the complete responders, if you follow them out to 5 years, at 5 years, you could guess what percentage of those who continued to be complete responders had persistence. And the answer is, it's a coin flip. 50% had persistence. 50% of those who had complete responses that lasted for 5 years, some would say that's a cure, did not have persistence at that point. So maybe it's the area under the curve that matters for the pharmacokinetic profile of our therapy. We're exploring that as one question. Maybe it's the Cmax of we just don't know yet. We are collecting all of the data, and we have a terrific translational research team and effort that is truly comprehensive, because we believe that we have a multi-year lead over every other company that would want to pursue this after we declare our first wins on efficacy. And the best way to expand that lead is to really understand the biology in humans in order to optimize the approach that we take to our MuSK program, as well as to our PLA2R program, and all of the others that will follow, both the announced and the unannounced. So we invest pretty heavily in understanding what's going on and why. We can't answer the question yet whether persistence is needed, but it sure does feel good to have it.
Right. I'm sure. It makes sense. So maybe looking a little bit more broadly or further ahead towards the potential regulatory path there, how well defined is that in this indication, and what might be considered provable endpoints in MPV?
It's interesting. So the answer is, there's risk. We don't know. What we do know is just based on observing others in the field, not in autoimmune CAR-T, but in CAR-T more generally. And we understand what FDA has accepted and what they've guided us on with regard to our clinical trial design. So I want to highlight for everyone, this is an outpatient study. It was our belief, it was our advisor's belief, and our SAB, very knowledgeable, experienced, well-informed group, as well as our investigators' belief, which FDA agreed with, that this should be a study that allows for outpatient administration of the CAR-T therapy. I think that suggests that all of us believe safety will be just fine. Now, you don't know until you know, and we continue to explore the doses. When we went to the IND filing process, which was accepted in 30 days with not a single change in anything that we had proposed in any preclinical, clinical, or manufacturing sections, FDA did make a comment. They strongly recommended that we consult with them on our Part A data before proceeding with Part C. we don't make a big deal of this. We called our trial a phase 1 trial. It's obviously in patients, and others would want to get credit for being in phase 2 or whatever it is, but it's a first-in-man study. And we thought FDA would want to know more just about the safety profile before we would proceed to Part C, which is the expansion phase of the study. But no, we requested a call because they said, this is not a clinical hold situation. This is just, we're giving you our best advice based on our experience in the field of CAR-T and gene therapy when you're going after a target that you can engineer against. And on a call that they accommodated for us within 48 hours of our request during the IND filing process, they came on the call and said, look, it's not a clinical hold issue. It's our advice that you it's our strong advice that you consult with us on your Part A data, because if your Part A data demonstrates good safety and a strong efficacy profile, then your Part C design, which is currently open label, needs to consider incorporation of a comparator arm in order for this trial to either be the label or to be a part of the label on the efficacy dimension. Having no comparator in Part C ties their hands as to how quickly they can move to get the treatment to patients. And therefore they were recommending, and on that call we agreed, we will submit our Part A data, and they suggested that they would respond in real time rather than a substantial delay of weeks or months in order to allow us to continue to enroll and advance this trial. So we don't know what the path to approval will be. We know that if the data are very strong on safety and efficacy, there's the possibility of incorporating another arm in Part C that would allow us to demonstrate efficacy against some standards of care that would be acceptable. And beyond that, we simply don't know. We do have orphan and fast track designation in hand, and we'll continue to pursue all of the regulatory opportunities to interact and to accelerate to get this therapy to patients if it's safe and if it's effective.
Great. And then another kind of bigger picture question on the program, in terms of the evolving landscape of the available and therapeutics that might be in development for pemphigus, as well as potentially other B-cell mediated autoimmune diseases. How do you see the DSG3 CAR-T program fitting into that landscape? And is there anything that you can learn from some of the misses that we've seen in this space recently?
So I didn't hear the first portion of your question. It broke. Could you repeat it?
Oh, yeah, sure. So in terms of the landscape of available therapies and those that are in development, as it continues to evolve, how do you see the DSG3 CAR-T program fitting in and relative to what's available, and how that might change in the coming years?
Yeah. So our view is we're not competing against any particular therapy existing or in development. We're competing against the disease. And what we're doing is unique in our view. We're trying to cure patients and give them deep, durable responses. When you think about the portfolio of targets that we go after, they are all pure B-cell mediated autoimmune diseases. They are clearly targets for rituximab. In fact, response to rituximab is often one of the things that we find comfort in. If rituximab works, then clinically it must be that the B cell is meaningful, at least. The FcRn class of drugs offers a lot of promise to many of these indications, and we think that those are all great advances. Rituximab expanding its label, the FcRns hopefully will be approved. At the end of the day, we're simply trying to cure disease. And annual administration of therapy, recurrences and relapses with side effects, with steroids, those are, in our view, all just need to be eliminated. We need to move ahead to a place where these patients don't have to live with their disease. We want them to live without their disease. And that's what we're shooting for. So where we fit or don't fit, the only indications we have so far Our current pemphigus trial does not require severe disease, does not require minimal, it could be any form of the disease, any severity of the disease, I should say. And it also allows for any patient, whether relatively newly diagnosed or with the disease for many years, to all be enrolled in this trial. And the reason we did it that way is that we don't know where we're going to have the best efficacy profile. So we need to try all patient types. If efficacy is persistent and constant across the portfolio of patients that we are seeing enroll in our trial, all of whom had to fail one therapy only, could have been steroids, could have been rituximab, could have been anything. As long as they have one recurrence, they can enroll, and with documented disease. And the addressable market for our therapy might well be all of those patients. If you're a payer, if our safety and efficacy profile, our safety, we have some insight, our efficacy, we haven't announced anything yet. If our safety and our efficacy profile are compelling, as a payer, you have a choice. As a patient, you have a choice. Do you want a cure that's a one-time event? Or do you want an annual therapy with the assurance that you'll need it again next year, that you'll have to pay for it, or somebody else will have to pay for it, and that you'll have to bear the side effects and expect that you will have a recurrence most of the time? Those are questions that only patients and physicians and payers are going to answer. Our job is to deliver the data to the best of our ability, and that's what we're focused on doing. It's important that this is delivered as an outpatient. So when you think about the value proposition, CAR-T therapy in today's marketplace, in the US, there are fundamentally different paradigms for reimbursement of CAR-T therapy when administered on an inpatient versus an outpatient basis. So we didn't design our study to be an outpatient-oriented study for that reason, but it sure is a good side effect, if you will, of the design, that the therapy could well be administered on an outpatient basis. The value proposition is substantially more interesting when administered on an outpatient basis. And frankly, any safe therapy that is effective, is an interesting value proposition.
That makes sense. Maybe just one last question on the mechanism and biology here related to the DSG3 program, as well as the other programs in the pipeline potentially. But from a biology standpoint, is there any reason to think that patients who don't respond to rituximab or are refractory to rituximab might be less likely to have a good response with a cell-based approach to target B cell, to target the pathogenic B cells?
Yeah, it's really interesting. So this has actually been studied. So patients who have recurrences on rituximab, the recurrences that they experience are always, and that's a really big word, a very extreme word, always found to be caused by a clone of B cells that was previously present during the first exacerbation. What that suggests is, as we know, rituximab does not get into the secondary lymphoid tissues and into the places where these B cells are going to hide. And it can't. That's why it's not monotherapy for cancer. It doesn't eliminate all B cells from all compartments in most patients. So these recurrences are due to pre-existing clones. By contrast, CAR-T therapy in cancer can be used to eliminate all of the B cells in all of the compartments of a patient and can do so durably. And so our belief is that this platform, which is borrowed directly from the early work that Penn did on Kymriah, applied now in a very specific and targeted way, should be able to deliver deep, durable responses in patients that are profoundly better than and different from everything else in development or on the market.
Got it, great. And so in the last few minutes before we wrap up, I'd like to ask if there's any other comments you'd like to make on read across from the DSG3 program to the myasthenia gravis and the membranous nephropathy program.
There are far more similarities between those three programs than meets the eye. They are all structurally similar targets, obviously completely different in the sense of specificity of our therapy, but very similar products to design for us. The first two, we already know FDA has guided. have preclinical programs that are nearly identical in their design. So we know how to do this, and we are executing, very efficiently, very well on those. The read across in the preclinical extends now into the clinical. Whether and how we fractionate the CAAR program, what dose we might start at or advance to, the impact of soluble circulating antibodies, the nature of the response that we might expect. There's so much read through. It's really profound. It is very much a lock and key kind of a system. We have a business end, a functional end of a CAR that is already the same one that's used in Kymriah, but now we have a targeting domain that changes from one product to the next to the next. The preclinical programs are going to be very similar, we expect. We haven't yet confirmed that with FDA on PLA2R. That'll come later this year. The clinical designs and programs are going to inform each other profoundly. The manufacturing is not identical, but sure is very similar. The Musk program we're going to be manufacturing in partnership with WuXi. We have dedicated facilities and dedicated people for our program contracted with WuXi for that purpose. And if the DSG3 pemphigus program evolves to become a commercially directed program very quickly, quicker than we would expect, WuXi would be a go-to option for us for commercial supply that could be used within the Part C of our existing study. So there's so many similarities that give us such leverage from the first program into the second and into the third, and then even beyond into deeper in the portfolio. Again, we like our odds on a safe platform of therapy in terms of the biology, and the leverage that comes across this portfolio from the first in man experience is, I think, as good as any company that I know of.
And so from a strategic standpoint, what is your approach regarding partnering to develop the platform or the target indications? For example, things that might not make sense to develop internally.
Yeah. Look, I know we have a product in the clinic. We have a second one coming. We have a third one that we've talked about moving pretty quickly. We are still fewer than 50 employees. We, in the past few quarters, have spent $10 million or less in each quarter. About 10 million or less in each quarter. We are an incredibly efficient, cell therapy machine directing ourselves to autoimmune disease. When we think about partnership, our number one question is, can you help us achieve cure for patients better or faster than we can do it by ourselves? If yes, then there's something to talk about. If not, then we need to see more data before we would want to, engage in partnerships that would, have to be value producing for patients and for our investors. And so we're, obviously in closing, very well funded. If everything is moving full steam ahead, we're funded at least through 2022. If things go sideways, the best and fastest case turns into a case of, our runway gets expanded into 2023. So we're just very comfortable that, we're executing in a way that is, smart and ready for that which we don't expect.
Got it. And so before we wrap up, I just wanted to ask, from your perspective as Cabaletta management, what, if anything, do you think investors are currently missing about the Cabaletta Bio story?
Look, if I was a great investor, I wouldn't be running a company. I'd be an investor, or I'd probably be on an island somewhere, but that's a different discussion. So I don't claim to know what investors are missing. I do know this. I do know that we, unlike the vast majority of earlier stage biotech companies, we are using a proven platform of technology. We are not changing it in any way other than the target that it's going after. And we're not going from B cells to solid tumors as CAR-T companies have in the past. We're going from B cells, 100% of them, to B cells, only 1% of them. We're going after a different target on that B cell. I like our odds to win. I just profoundly better odds than I think the vast majority of companies out there. And there are many companies that are valued in the billions and have yet to enter the clinic on the promise that they will cure all diseases that you and I fear. We're just trying to cure autoimmune diseases that are B cell mediated, that include myasthenia gravis, membranous nephropathy, pemphigus vulgaris, the hemophilia antibodies that prevent factor VIII from working, and many more that are behind it that we're advancing in an incredibly efficient platform that has a great read through across the portfolio. I don't know what anybody could be missing with that, but that's what we're doing.
Sounds great. All right, I think we're at the end of the hour, so I'd like to thank you all again for your participation in the great discussion.
Yeah, thank you. Thanks for the opportunity, Geulah, and thanks for investing the time.